So yes, myrtle rust infection of an endemic rainforest tree across a forest edge gradient in New Zealand. We've heard a lot about Lophomertus ballata, ramarama, most affected species. It's a 6+ metre tall tree, but it's usually smaller. Most people would probably describe it as a shrub if they hadn't actually seen some sort of giant individuals. You'd usually find it in wetter areas if you were wandering around a forest, riparian kind of spots. And you also often see it at forest edges. And this is kind of how the study started. It was from observations of myrtle rust at forest edges and sort of questions about whether or not there is more infection at the edges, or maybe it's just that people tend to be at edges, at track sides, and so on more often. So it's actually just more, it's observed more there, but it actually occurs kind of throughout. So we decided to set up a study to look at this. A study that looked a little bit like this. So, pointing at this one, so we set up these transects running from sort of a forest edge, which is this black line here, outside. In our case, it was pasture, but just some sort of non-forested matrix was the idea, and then heading into the forest. In this case, we set up a 60-metre transect with these kind of 5-metre subplots dotted, extending throughout 5 by 5 metres inside 15-metre what we called zones. In the middle of each of these zones, we had temperature and humidity sensors. And then in each of these 5 by 5 metre subplots, we measured every single Lophomertus ballata individual within them, including their basal diameter and height, to get some idea of their demography. We looked at plant community composition using vegetation releves or recces. So it's basically just an assessment of what plant species you have co-occurring, in those subplots and their abundances. We took a measure of canopy cover using a Lycor LAI machine. And then also in each of those subplots, we tagged 3 individuals for further monitoring for disease assessments. And we selected these as the tallest individuals that were less than 2 metres tall and greater than 0.3 of a metre high. So we didn't always have 3 individuals in a subplot that fit with those criteria, but usually we did. And we went back and revisited to look at disease load, and also we did some microbiome studies, but I won't be presenting that today because those data are still being analysed. So we set up 3 of these transects in November and December in 2020. And actually, since Lophomertus ballata didn't tend to actually go right to the edge in these plots, we didn't actually start them where there weren't, in places where there weren't any Lophomertus ballata. So it's not strictly sort of an edge study like I described anymore, but instead we were sort of focusing on relationships between light availability and distance to the edge and so on to make assessments about the effects on disease. So there were 3 plots within which there were 72 of those 5 by 5 metre subplots, and just almost 1,000 Lophomertus ballata individuals were in those subplots, of which we tagged 146 of them for further study. So this is just typical, an image of an area typical to these transects. That's the brighter green tree that you can see, shrub that you can see there is Lophomertus ballata. Other co-occurring species, we had Kaikatea, which is white pine, is a common name, but lots of tree ferns and so on. So typical kind of temperate rainforest setting in New Zealand. We did 3 disease assessments in February 2021, 2022 and 2023. So this was at about peak infection. Michael Bartlett from Scion and his colleagues have a tree that they monitor quite intensely, really close to where these ones are. So we sort of use his data to decide when to go in. And for each of these assessments, we, on those 146, I think it was, or whichever number, individuals, we inspected every single leaf and scored them as either immature, because it's only the immature leaves that are susceptible, mature or dead, and then whether they were also infected or uninfected. And it would only be the immature leaves technically that should be infected. And then we also looked at immature stem sections and buds, so other sort of young tissues that were associated with them. Then we also counted the number of infected and non-infected Lophomertus ballata individuals within a 1.5 metre radius of those focal plants. And as I said, we have 3 years worth of data to look at how those numbers change over the years. So here are just some some sort of overarching numbers. In 2021, those individuals that we were looking at had had 37,500 leaves across them. When we went back a year later, those same individuals only had almost 29,000 leaves. So that's a huge decline, which is an interesting thing to note right there. And then another decline this year when we went in February down to 26,500 leaves. So before I talk too much about the results, what I'm presenting here is just a preliminary analysis. We're actually redoing some of it now, so there might be some changes by the time these are published if you see this come up again. And also, most of the results I'm presenting are from the first disease assessment that we did in February 2021. But anyway, what we're looking at here is a mean percentage of the immature leaves that were infected on our tag plants across the 72 subplots. And hopefully you can see that there are definitely differences in infection level, with deeper red colours indicating a higher level of mean infection of individuals. But at first glance, you might not see that there's a particularly obvious pattern to it. As I said before, we're initially interested in that forest, that kind of edge effect, but just sort of looking at it, the transect on the right here, maybe there's higher infection closer to the edge, but this one here certainly seems to have infection all the way down. So you really need to start doing some modelling to start looking at the patterns behind this. And this is a bit of a horrendous plot, sorry, but we did find a relationship between disease severity and distance to forest edge, but what we did find was that it was mediated by canopy cover. So this is a modelled surface of an interaction between distance to forest edge and canopy cover, with myrtle rust infection on this sort of y-axis here. The model starts getting a bit out of control at the top here, which I don't think is biologically reasonable. So I just sort of tell people to imagine that part of it's flat. This is part of what we need to start teasing out with further analyses. But if you just look at these two axes here first, so this is level of myrtle rust infection, and then you've got distance to forest edge here. And if you look at the line here, then you do see a pretty obvious decline where you head into the forest edge, you get a drop in myrtle rust infection. But the key point there is that that's only when you have low canopy cover. So see this axis. When you have high canopy cover, that relationship drops off. So you've got high infection regardless of where you are along the edge. We put our thinking caps on with that one, and we thought it might have had something to do with humidity. So if you've got higher canopy cover, then you might have sort of a wetter and more moist environment. It's always moderately warm there, but it might just be that higher humidity that you have with higher cover. That means that the edge effect sort of drops off and you get infection regardless. There are a couple of other aspects to the model as well, some other covariates we put in. The number of large trees in the subplot. As you get more large trees in the subplot, infection goes down. So this could be analogous with sort of more advanced forest, larger trees, more intact. Maybe a little bit counterintuitive to the humidity effect I was talking about before, but if you think back to that satellite imagery that I showed you before, it's pretty much all got quite high cover. So you're talking about, is it high or is it really high? And actually, when you've got really high canopy cover, it might actually be a more complicated and recovering forest with smaller stems, which has that higher infection. Another one, and this is probably kind of obvious, as you have more infected, if there are more infected neighbours around you, then you tended to be more highly infected. And then just the final plot that I'll show you now is our first kind of assessment looking at the change in disease, and this is between year 1 and year 2, so 2021 and 2022. And we found, so that area in the red, that's an increase, and below that is a decrease. So as you head further into the forest, we actually noticed a decrease in the amount of infected leaves, and individuals close to the edge had an increase. But there are some nuances that we still need to tease apart with that one, because there were quite a few individuals that didn't actually have that much infection, and they didn't have many leaves either. So we suspect that they've actually just lost their leaves. So that change in infection is probably not the best metric for whether you're badly infected or not, because they may just have not had many leaves on them due to myrtle rust. That's it for me for this one, with some acknowledgements from Naruhine and Taranaki Iwi, who helped us with this, landholders and DOC, and heaps and heaps of people who helped us count all of those leaves and also helped us find the sites and so on. Cheers.
Any questions?
Sorry, you probably already mentioned it, but just to remind me how you measured canopy cover?
It was using a LICOR leaf area index device, I suppose. They're often used for crops, and it gives you a leaf area index, which is sort of a surrogate for canopy cover. And we did 9 measures of that, kind of like if you had a 9 on a dice within each of those 5 by 5 metre subplots.
Thanks.
We've also got another measure of cover that we're playing with, that Releve vegetation data that I was talking about. We've got percentage of vegetation cover in tiers as part of that. So we're also sort of wondering about interrogating those data.
And is the results of this in press or anything yet?
No, still being, the analysis is still being finalised, and we'll be writing it up over the next year or so.
Thanks.
Thanks.
Any more? Do you know what the other species are in each of the plots?
Yes.
Cool.
Yeah.
You'll analyse that as well?
Yeah, we've been looking at, there are so many ways you can analyse these things. We've looked at sort of number of co-occurring myrtaceae and their abundance, kind of along the themes, I suppose, of the spillover work that others are doing at the same site. And we've been looking at, actually it was something that Roanne mentioned when we were presenting this, looking at the sort of the structural diversity. So you can go, okay, is there lots of abundance and lots of tiers above it, which tells you about what kind of vegetation it is, or is it something that's, you know, dominated by one tier, which might be a more advanced sort of forest type, and within that you've also got different species compositions, so there's quite a few ways to take it.
Cool. Awesome, thanks.
Yeah, I'll go on. Does your humidity or temperature stuff account for dew? The stuff I looked at, I think under the canopy, dew was a big player in infection.
Yeah, we don't have a good measure of dew, but that might be higher under a closed canopy. Yeah, that's a good point.